Good afternoon. I'm Robert DeFilippi, the co-founder and co-director of the Center for Innovation and Change Leadership at the Sawyer Business School, and it is my pleasure to welcome you to today's third annual Global Leadership and in Innovation and Collaboration Award activities. I call it GLIC because that's too many words. The GLIC program. I want to just say a few words on our center and a few brief introductions to some of those affiliated with the work of our center. The Center for Innovation and Change Leadership was created in 2004 to promote innovative and collaborative solutions to business and social problems through a variety of initiatives featuring research, teaching, consulting, and service to the community, of which today's event is one example. A brochure is available in your program to provide you an overview of some of the details of our center. Please review it at your leisure. Next, I'd like to just thank a few folks in a few groups, not by individuals, but by a group, and ask them to stand so that those of you who are not part of the center might know who is affiliated with the center in case you might like to chat with them a bit during our reception to follow. So I first want, our center includes faculty fellows from 5 academic disciplines of the Sawyer Business School, and we have several faculty with us today, faculty fellows who are here. So I'm wondering if those faculty who are fellows of the center might take a moment to stand so you can be recognized. Rick? Yeah. Anyone else? Arnold is here as well. Arnold, thank you both. Next, we're also, very ably assisted by a wonderful advisory board, and I'd like to ask those members of the advisory board who are with us today to just stand and be recognized. Richard and Larry Stiebel, Bill, Pat, who's doing double duty. Not only is Pat a board member, but she's also providing videography services for the day. Thank you all. And Ed Torino, who is a member of our board. Thank you very much for joining us today. And lastly, this may be double counting, our nominating committee. There's a group that's responsible for vetting our candidate organizations to honor, and if I could just take a personal, it was Larry Stiebel, who I think brought to the nominating committee's attention the very worthy work of the Accelerated Cure Project. So I want to take a moment to personally thank Larry Stiebel for bringing ACP to our attention. Are there any other nominating committee members here today? Lisa, right? Lisa O'Donnell, please. Member of our nominating committee responsible for selecting the wonderful organizations we've been able to work with these past 3 years. It's my hope that you'll take the opportunity again to chat with a number of the people I've just identified during our reception that follows today's events. Now I want to just provide a very brief overview of today's event. This annual GLIC activity is really a day long of it. It began at noontime with Robert joining a group of faculty and leaders of the business school over lunch to talk about some of his work, and it was then followed by Colette Dumas interviewing Robert in our television production studio so we could immortalize both on YouTube. And more recently, we just had a little photo shoot where we are hanging in our Innovation Hall of Fame gallery a lovely photo, lovely full-length image of Robert McBurney next to our other award-winning recipients. But the main event is now. And
Insight is a strategy and innovation consulting firm founded by Professor Clayton Christensen of the Harvard Business School. And in the 90s, Professor Christensen, through his research, identified patterns of corporate strategy where dominant incumbent firms leading their industries suddenly found themselves losing customers to small entrants who had a way of nailing those customer needs that the incumbents didn't. And a lot of those incumbents ended up ceding their leadership positions, losing customers, and indeed going bankrupt. So Insight helps companies of all sizes really understand what innovation is all about and how to put these principles to work to grow and also defend their positions. So for this presentation, as mentioned, we're going to talk a bit about 4 key best practices of innovation that we work with companies on and how ACP exemplifies them. The first key piece we'll talk about is the job to be done. All value-creating innovations start with an opportunity, some pain point, something that they address. Then they create a business model wrapped around a technology. So you're going to try to come up with the technology or the solution to address it, but wrap it in a business model that allows an organization to create and capture value. Next, you're going to keep an open mind, make sure you embrace innovation, but open innovation and collaboration, because it's very rare that we all have the one right answer. And then finally, you're going to make sure that you test and learn. because again, as we'll talk about in a few minutes, 90% of all successful ventures started out with the wrong strategy. So right at the beginning of any company, the one thing you know for sure is you're wrong. You just don't know in how many ways. So the job to be done. Ted Levitt was a professor at the Harvard Business School for nearly 30 years, a professor of marketing. And one of the stories he liked to tell was that people don't go to a hardware store to buy a drill. They go to buy a hole. The drill is just the best way to get that job done. So the job to be done in this case is a fundamental pain point or frustration. It's a challenge that somebody's trying to get done. And the greater degree that frustration is important, unsatisfied, and widely held, the greater the opportunity for innovation. Now, I don't have to tell anybody here the opportunity created by the challenge of multiple sclerosis. So there's over 400,000 individuals in the US with MS today, and over 200 added per week diagnosed with the disease. Over 2.5 million individuals have MS around the world. And on average, patients with MS live 5 to 10 years fewer than those without. So talk about importance, unsatisfaction, and widely held. There's no cure. You can cope, but there's no cure for the disease. This is the foundation with which Art, right there in the center, founded ACP. Frustration with the fact that there really was no cure, and all the research efforts that were taking place were largely siloed and very small scale, which was a real challenge for something the scale and the complexity of MS. So the pain was felt by patients, was felt by researchers, and something had to be done, and that's where Art stepped in. So number one, a great opportunity for innovation. So now the innovation itself. Now the iPod, as we all know, was really successful. It matched a great technology, MP3 compression, with great hardware design and a great operating system. But it didn't truly succeed, didn't truly begin to change the industry until it was wrapped with a great business model, which was iTunes. A razor and blades model just like Gillette. Sell the device, sell the songs for cheap. And that locked everyone in, and that created the atmosphere we have today for Apple. So most successful innovations truly wrap a good technology in a great business model. As mentioned, the complexity of MS demands large scale, demands multi-factor analysis, moving away from individual siloed research efforts, which have been the hallmark of the disease. And so ACP comes in, creating two key things: the repository, the biobank of samples, and also the cure map, both of which are open, both of which are collaborative, and at a basic level are great technologies that enable MS research to progress in ways it hasn't before. But the further key was wrapping those good technologies with a great business model. And that's that not-for-profit approach, which allows funds to be raised to drive MS research at a scale that's never been done before. It allows the positioning of administration to cover these two great technologies, which would be very hard for individual researchers to do. And the publish or perish sort of penalty doesn't exist. So these good technologies aren't necessarily the things around which research papers could be written, yet this is something that ACP can do. So again, a good technology wrapped in a great business model. It's a great example. So next we have the approach. The guy in the middle is a British economist, John Jukes, and he did research on innovations in the 19th century. And what he found was that 46 of the 58 major inventions in the 19th century started off in the wrong place. So a pair of musicians actually came up with the first instance of Kodachrome film. A watchmaker playing with brass for the watches came up with the process for continuous casting of steel. And the originators, the entrepreneurs who created the first jet engine, were actually told by reciprocating aircraft engineers and inventors that it was useless. No one would ever want it. The key here is openness. You never know where the answer is going to come from. And as I've talked about already, that's a key component to ACP's strategy. A lot of scientists, a lot of efforts out there, we don't know where the cure will come from or what pieces will make up the cure, so be open and collaborative. So not only is that a hallmark to ACP, it's a hallmark to what Robert does in his leadership of the organization. He emphasizes the tenets of being open and collaborative, building bridges and relationships, making connections among scientists and sharing data, and empowering those individual scientists that he'll talk about in a moment. So that's a key pillar 3. And finally, the journey. So as I've mentioned, the only thing you know when you start a venture is you're likely to be wrong, you just don't know in how many ways. And so the best thing you can do is take a path that mitigates risk. So rather than venture forth to what we call the edge of the basketball chart up there, be iterative. Conduct small experiments. Learn from failures, learn from those experiments, and redirect. That's the only way a cure is going to be found from, a cure for MS will be discovered, given its complexity, given all the factors that are involved. Not only, again, is that a hallmark of ACP's strategy, it's something that Robert talks about often, in terms of valuing intelligent failure, valuing individuals who have experience learning from failure, and actually put that to work. So that's tenet 4. So these are 4 key best practices that you can find in virtually every organization out there that's come up with any game-changing innovation. You start with the job, you wrap a good technology with a great business model, you keep an open mind, and you always test and learn. And ACP is a great example of that today, and Robert will talk about that a bit more. So, Robert?
Thank you. And Suffolk University generally for what is a remarkable honour for the organisation that I've been CEO of for just 4 months. And so I'd actually like to begin by asking those people in the room that are associated with the Accelerated Cure project to stand up so that you can all be recognised. So I have to begin by praising Art Meller for his insight in starting an organisation that was so compelling to join that I couldn't resist under any circumstances. And the interesting thing about the organisation is that it was started not by a biomedical researcher, but by a high-tech entrepreneur. A high-tech entrepreneur that, in terms of the way we get things done, was just so far ahead of traditional biomedical research. The concept of open source, for example, is so rare in biomedical research for a whole variety of reasons. But the concept of open source has tremendously accelerated the software industry. And so thinking about how I could do something new to create a biomedical research activity is something that I want to talk about today, because although it's 10 years old this year, Accelerated Cure project is really still just at the beginning. And we've done a lot so far, but I think that there's a tremendous amount to be done going forward. I'd like to show this picture to begin with, and you'll see that I'm going to show mostly pictures and talk about pictures. So this picture is Anna Peabody with her parents. Anna Peabody is a multiple sclerosis sufferer who thinks that what Accelerated Cure project is doing is fantastic. And it's not that she thinks that Accelerated Cure project is fantastic that I want to emphasize here, but I want to emphasize that Accelerated Cure project is all about bringing a cure to multiple sclerosis patients faster than looked like it was possible 10 years ago, and still looks like it's possible today. So I don't intend to spend a lot of time talking about multiple sclerosis. It's a very debilitating, degenerative disease of the brain and spinal cord. Part of the disease is caused by the person's own immune system attacking the nervous system. It shares features with rheumatoid arthritis and Crohn's disease and a variety of other autoimmune disease, but it's not just an autoimmune disease. It has 2 features. It has a relapsing and remitting feature. It usually begins with someone, for example, losing their sight temporarily because the immune system attack has overcome the regenerative process. And then the regenerative process takes place and the sight comes back. And this usually is very disturbing. And it's the beginning of what turns out to be a rather long investigation of what exactly it is that this patient has. Alex mentioned some of the figures. Basically, it comes down to about 1 person an hour in the United States diagnosed with multiple sclerosis. there are treatment options. 10 years ago, there were very few treatment options. Today, there are 8 approved drugs. So there's been actually a remarkable progress there. But unfortunately, those drugs only deal and only partially deal with the relapsing remitting part of the disease and don't focus on the progressive disability, which ultimately is the thing that shortens lifespan. And that's primarily because we don't understand what the mechanism of the disease is. So we don't understand what the relationship is with this remitting relapsing and the progressive disability. Because there are 8 drugs, and there are lots of different flavours of multiple sclerosis, the treatment strategy tends to be trial and error. And I mentioned that there are 8 drugs now. There's a tremendous amount of activity in the pharmaceutical industry at the moment to produce new drugs, most of which are just more drugs for the relapsing remitting phase. And so we're going to have 12 drugs. Physicians, in a bizarre way, physicians are now faced with a lot of choice and are beginning to tear their hair out, to work out what is the right treatment to give to the right patient at the right time. So there's a whole area that is being opened up, and it's a very costly disease to treat. So you see this figure here of $80,000 per relapse avoided. Generally, the average number of relapses per year that a patient has is about half. In other words, 1 relapse every 2 years. So essentially, to avoid that relapse is about $80,000. It actually ranges up to about $140,000. And there are very few guidelines that physicians have to know how to treat the disease. So, sorry, I keep pressing the wrong one. To solve these problems, we simply need a better approach to research that helps us understand more about MS and directs us towards a cure. So we need lots of things from research. We need to understand the causes and mechanisms of the disease. Despite the fact that there are drugs on the market, they mostly approach suppression of the immune system in one way or the other. And they're related to the drugs that are used currently for rheumatoid arthritis and a variety of other conditions, which we don't know the fundamental cause of, but we know something about depressing symptoms. We need a lot of other information, which I won't go into here. We actually, at the bottom of this list, we need much, much more. And so, when the organisation was founded, and I think ARC would say the same thing today, we don't need just to raise another $200 million to foster multiple sclerosis research. We need to do something that's different, something that will transform the way that research is done. So I'd like to talk about these 4 images in my presentation and build the story about Accelerated Cure Project around these 4 images. It's a picture of the telegraph and an iPhone, a picture of a cafe, a postage stamp, and a satellite view of Antarctica. So why talk about the telegraph and the iPhone? Well, the answer is that that's a perfect example of innovation. Not just one innovation, but a succession of innovations over time that have transformed the way we live. I bet there's no one in this room that can remember using the telegraph. That's the way it is with innovation. Once an innovation comes along, sometimes you can't remember what life was like without it. And so that's what Accelerated Cure Project is trying to do in multiple sclerosis. It's trying to change the paradigm in research so that we don't remember what traditional academic biomedical research was like. It's a tall order, but I actually think we've gone some way down the line. Now, how do you make innovation happen? That's one of the key. Once upon a time, we thought that we didn't actually know how innovation happened. You know, innovation just sort of stuff, just whoosh, came up there. But I think if you look at Steve Jobs' career, you'll realise that there's something more systematic that happens that makes innovation happen. And I point to this restaurant. This is the Grand Cafe. This is in Oxford, in England, and it's the site and the building of the first coffee house in England in 1652. Schultz had nothing to do with this one. So why do I show a coffee house? Well, coffee houses, if you've ever read any of the work of Stephen Johnson, you realise that his position is that coffee fueled the Age of Enlightenment. Because what happened in coffee houses is people came together and talked about stuff, and they refined their ideas. And by refining your ideas, you make better ideas and novel ideas, and you have the musicians that create film and such like, because they shoot the breeze. Well, so why did coffee make a difference? The answer was that prior to that time, the drink of choice were alcoholic. You didn't drink the water, you drank beer, or something like beer, or something like wine. Now, wine and beer are a great stimulant for a short period of time, and as I like to say, then you start singing. And you don't remember any of your ideas. Coffee changed that. Coffee's a stimulant, and discussions held under the influence of coffee were much more productive. And within a couple of years in England, there were hundreds of coffee houses, a couple of hundred in London, and our banking industry, insurance industry, foreign trade, and a variety of other institutions actually grew out of the coffee houses in London. So, what relevance has this got to ACP? Well, one of the things that we've been doing in addition to creating the Cure map, which brought together a lot of ideas, is that we've been creating dialogues between people from different backgrounds. Remember, it was started by a high-tech entrepreneur. And so we have MIT engineers who think about multiple sclerosis today in our family. That's a strange thing, that doesn't happen that often. But we're going one step further. And to give you a little view of the future, we've been very fortunate to come into a collaboration with the Mass General Institute for Neurodegenerative Disease, with some funding from EMD Serono, that has allowed us to begin to build, for a launch in February, the Multiple Sclerosis Discovery Forum website. The Multiple Sclerosis Discovery Forum website will be the 21st century MS Cafe. It'll be the place that people come together to shoot the breeze in the ether. It'll be open 24 hours, and it's part of the Accelerated Cure Project's approach to open access. So we want people to share ideas. We will provide information on that site, we'll provide access to resources, and we will promote things like Innocentive. Do you know what Innocentive is? It's this website that poses problems that people can submit answers to. And I think there's another one called Top Coder. So we want to make the 21st century MS Cafe a place where people develop ideas, where they post problems, and that it stimulates and advances the field in a very open way. Second image is a postage stamp that happens to have Margaret Mead on it. And of course Margaret Mead is famous. Margaret Mead, as you know, is an anthropologist who studied societies and how people live and organise themselves and achieve the objectives they want to achieve. And one of her most famous quotes is, "Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has." And so when all of these ideas are being generated by people that perhaps are not even well-established researchers in the multiple sclerosis field, how are they going to get an opportunity to explore their ideas? So idea generation is one important thing, idea exploration is another. And so that concept led to Accelerated Cure Project developing the ACP biorepository. We thought, wouldn't it be great if instead of having to conduct a clinical trial to get 400 samples to study some molecular components of DNA or otherwise, wouldn't it be great if you could just submit a proposal to some organisation that had all of these samples and they could give you the samples? And so Art Meller decided ACP should have a biorepository. And that started in 2006. It currently has 10 clinical sites around the United States, so it's got quite a bit of ethnic diversity and geographic coverage. It currently has almost 3,000 samples. But most importantly, these samples are collected with the highest quality, and they're also associated with a 52-page case record form of everything about the patient from which the sample was obtained. These samples are aliquoted into small volumes and can support hundreds of studies. The key is that access is given to scientists from any institution worldwide. We currently have work going on in Germany, Israel, Australia. Did I say Australia? I haven't noticed the accent. And in the United States. There's a very streamlined application process. Now, if I was a researcher and I wanted to generate 400 samples, blood samples from multiple sclerosis patients in a well-controlled fashion, I could tell you it would cost about $2,500 per sample, because that's actually what it costs ACP to do it. Which means 400 samples would be $1 million grant that you would need. Well, this is, firstly, many small groups with novel ideas are not going to get that $1 million from the NIH, simply because they're unknown or they, for a variety of reasons. So obviously this provides the potential for a low-cost access. We provide those samples at $50 each, plus some additional coverage costs. So 400 samples now cost $20,000, which is certainly easy within your research realm. The key is that if you take our samples and you do some analysis of those samples, you have to agree within a certain period of time to deposit the data back in our database. We'll give you the time to protect any intellectual property. We don't want to own the intellectual property, but we want to give you the time to do that. But it's not a long period of time. You've got to make a decision and do something about it before you get the data back, because we want to accelerate the process, not hold it back. So we're currently supporting over 50 studies out of this database. There have been 15 million plus data points returned, and we're actually only in the very earliest stages of this process. About 16 publications, actually one available diagnostic test already out of this activity, and other diagnostic tests in development. So that's the second image. Basically what we're doing here is, if you like to think of the innovative young groups as squeaky mice, and you like to think of the established research groups as the lions, what we're doing is we're efficiently enabling the squeaky mice to roar. And that's basically the principle behind creating the repository. That's very much an exchange of samples for information, and you could call that a collaboration, but actually we're going beyond that. So one of the things that we had our annual symposium Saturday week back. We brought together many people from the 10 clinical sites that we interact with to pose them the question. At the moment we have a hub and spokes relationship with you people. We provide money and you send samples, and we have nice dialogues and we can talk to you. What say we create a MS clinical network so that we can chat about projects that none of us could do alone. So the resources of 10 clinical centres makes it possible to do much larger scale projects. So that discussion is very active at the moment. It really only started 10 days ago. But it's a great deal of enthusiasm, and you might be surprised to find there is not an MS clinical network in the United States. There have been some that lived and died for a variety of reasons, but there is no active MS clinical network right now. So there are no groups of people necessarily conceiving very innovative projects that could be supported by ACP's resources. The other thing that's moving very fast at the moment is the area of bioinformatics. Things like disease modeling, taking vast amounts of data and developing models of disease that you can then use to indicate new drug targets or things that will separate one group of patients from another that might help to answer the question, well, who is going to respond to any particular drug at any one time? And we're involved in a discussion at the moment, and it's a little bit premature to say this, but there's an organisation that's been created by Patrick Kennedy and other people called One Mind for Research that is focused on bringing together a lot of resources and new funding for research into nervous system diseases. Underneath that parent organisation, a group has been created called One Mind Bionetworks that's really interested in modelling neurological diseases. The first disease they've chosen is multiple sclerosis, and the place they've come to get data sets to put into their model is ACP. So we're really excited about these collaborations, both of which are not fully formed yet, but it's the sort of thing we're trying to push forward right now. So finally, David Blom is confused about this. What has this got to do with anything? This is a beautiful satellite picture of Antarctica. And right in the middle of the page there is the South Pole. Of course, it's not. You cannot see the flag. So why show you a picture of Antarctica? Well, the answer is this is the 100th anniversary of the race to the pole of Amundsen and Scott. And you may remember that, you may not remember, but you may be aware that there were quite different outcomes for those two attempts to reach the South Pole. Amundsen was successful. There was a lot of controversy around the way he was successful. Scott got to the pole, but never got back. There are a variety of reasons that Amundsen was successful and Scott was not. One of them was planning. Amundsen actually took a different path. And one of the things that one of the biographers of Amundsen said was that the most remarkable thing was not that he took many more dogs along in order to feed the dogs and feed people along the way, which was certainly an excellent plan at one level, but that he decided to take not the usual path, and that not the usual path was successful. But also, planning was successful, having a plan. And so I just wanted to show you some of ACP's 3-year goals to conclude. One of our goals is, once we've launched the Multiple Sclerosis Discovery Forum website, we need to gain the resources to keep it going for an initial 5 years post-launch. We actually have support for it to be going for a year post-launch from the original grant. We want to double the size of the repository. There are many reasons why we want to double the size of the repository within 3 years. One reason is the appearance of new drugs, so we want to get information about how patients respond on those drugs. We want to create new longitudinal studies so that in addition to finding out things about the causes and mechanisms of disease, we can find things out about the prediction of the course of disease based upon certain treatment strategies for individual patients. We're doing something quite exciting on the database side. We're redoing our database so it can handle much more data. It's going to live in the cloud. And in addition to that, we're building a patient portal into the database. So patients can self-report their outcomes. And we'll be able to gain a lot more data about the patient outcomes. So most multiple sclerosis patients don't see their physicians all that frequently, and we all know that when we go to see our physician, we're on our best behaviour. Oh no, it doesn't hurt. Oh no, it didn't. And what we want is we want the real outcome data to compare with the treatment strategies and to look at what mechanisms might underpin some of the symptoms of the disease. And so that's a very exciting part for us. And it's the sort of open source, open access approach. We want to do much more molecular analyses, and we need to gain support for more molecular analyses. There are some approaches that very few of the groups that we are interacting with can undertake, and we want to try and stimulate the generation of much more data that can be used by such groups like Up1Mind Bionetworks to model the disease.
Thanks for giving us an interesting talk. I have a question a little more global. I think it's great that you're doing this in the MS space, but we know that pharma actually has a really big problem with their pipeline. They are not developing drugs as fast as they would like to, not only for monetary compensation, but for helping patients. Do you see what you're doing as a potential model for pharma, and how would you help them if they wanted to do it that way?
Well, we actually are already helping them, because many of the biotech companies particularly, and pharma companies that are focused on MS, are accessing our samples as well, which is remarkable, actually. I mean, why should that be necessary? And the answer is that they didn't think of collecting samples like this. A lot of samples that are collected in the pharma industry are disease patient and disease patient treated with drug. That's what comes out of clinical trials. They never have the healthy controls. And so they can't make that comparison that allows them to come up with potential mechanisms and causes of drug targets. And so they rely primarily upon the academic community to do that. It's interesting. I don't know exactly how this would work inside pharma. But is there the need for more accelerated cure project models like this? Absolutely, yes. I was at a conference in Florida, the BioFlorida conference, earlier last week. And after I'd given a talk, which was in a session on how patient advocacy groups can stimulate research, a senior official from a major university in Florida came up to me and said, "Oh, have you branded this concept? Couldn't there be the accelerated cure project of diabetes, the accelerated cure project of rheumatoid arthritis, etc.?" I said, "Of course there could." She went, and walked off. So I think the reason I joined the organization is I think this is a great idea. And I think it is actually accelerating research. And it's likely to do more than that. It's likely to actually accelerate personalized medicine as well. But exactly how one would integrate it into pharma, I'm not sure, because as you know, culture kills strategy. And in as much as this is a strategic change, it does not mean that there are not pharma and biotech companies that are really supportive of Accelerated Cure Project, and I'm expecting more of that as the message of the success gets out there more. But integrating it in, I haven't really thought deeply about that, because I think that the culture kills strategy piece is a major barrier.
You've talked about, you've come up with one diagnosis thing that you can actually use. As you come up with activities that will actually be used, do you own them? What happens then, particularly under an open source kind of situation? Who owns them? How do they then get out into the field? Could you talk about that aspect of it? Because clearly that's going to be stuff that will be coming out in the next few years with you or with many of the associated researchers that are working with you.
It has not been Accelerated Cure Project's vision of owning any intellectual property to date. The way that intellectual property is created is that our samples are analysed, data is created. If that data can be included in a patent application, that's up to the organisation that has taken the samples. The data must come back to us. So it can be, actually the whole purpose of the patenting system was to provide a vehicle for, not keeping everything as a trade secret. So we're just practicing that model. And the way that these things come forward to products is that as a result of the intellectual property, then that is either moved forward by the organisation. We have small companies that take the samples. moved forward by the small companies that take the samples to the point where they're commercial products. We don't ask for any return for that, but some of the companies that we work with are generous in their donations to accelerated cure projects. So there's no direct link between any intellectual property. I'd like to say one other thing that's relevant to this, and it needs to be thought of very seriously in an activity like this. So in my position as Chief Scientific Officer of a couple of biotech companies, I chaired the intellectual property committees for about 15 years in total. And you learn a lot about intellectual property, the patenting process when you do that. What I learned was that filing a patent was an opportunity to spend $250,000 with no guarantee of anything. And a good friend of mine, Ashley Stevens, who used to be in the Community Technology Office at BU, has written articles on the success of tech transfer offices in universities and colleges. The answer is there are very few tech transfer offices that are self-sustaining. And one of the reasons they're not self-sustaining is all of the money they spend on patenting inventions of their faculty that never really quite go anywhere. Now, of course, there are some remarkable exceptions to that, but on balance, owning a lot of intellectual property and paying all of the costs associated with that is no guarantee of anything else but owning a lot of intellectual property and paying a lot of the cost to move it forward. So that's something that factors into ACP's thinking as well.
Other questions right there?
So thinking down the road, however many years it takes, what will you do when you find a cure as an organization?
I think we
Where do you
declare
go from
victory. No. Well, I think it will be incremental along the way. I mean, a cure, you know, if you think of what a cure is,
Yeah.
so a cure is not just a drug, because that drug may only be relevant for a small proportion of the population. And in addition to that, that drug has to be delivered in a healthcare system. And so I think there will be a number of small victories along the way that will gradually, and it's very interesting when you look up the definition of cure. So most of the definitions of cure, including the Oxford English Dictionary, which I found most surprising, doesn't talk about a complete removal of the disease forever, but in some cases for a prolonged period of time. And so it comes back as well to what the definition of cure is. What we'd like to do is prevent the disease, and that's included in the statement cure, although not explicitly said. So it could be that one day there is a vaccine that will eliminate the possibility of the disease from some subset of the potential patient population. And there is actually, as you probably know, a viral hypothesis that underpins the idea of the cause and mechanisms of multiple sclerosis. It has never been proven, but one of the ideas with a lot of autoimmune diseases is there's some infectious agent that is introduced into the body, which is then attacked by the body's own immune system, and that infectious agent has some structural features which are very similar to the structural features of the sheath on nerves. And so the body goes, oh, that's another one. But that has never been completely proven. So I think that multiple sclerosis is not just a single disease, and I think that what we're looking for is to begin to make incremental gains. And one of the diagnostics that's already available is actually characterizing the multiple sclerosis patient at a specific stage in the disease that then allows physicians to have a specific treatment strategy. So that's an incremental gain already. But I think there'll be many incremental gains, and we will declare victory when it's all gone.
I don't remember whether whether you said ACP actually does any research itself, But my impression after listening was that mostly you play a coordination function. It sounds like you've been involved in leading research directly in the past. Can you speak to the difference in dynamic between being a coordination function and actually leading the research yourself? And what that means as far as how the center is run and that kind of thing?
That's a very good question. There is a big difference when you're leading research yourself. You make a commitment to a particular hypothesis. And what we are doing is making a commitment to provide many hypotheses to be explored. So when someone sends us a proposal for samples, we don't critique that proposal for, well, at least at the, if it's anyway, we don't critique the objective of the proposal. We critique the design of the experiment. So we often come back and say, have you done a power calculation based upon what effect size you might see, and based upon the understanding of your measurement technologies, variability and things like, have you done that? What's your experimental design? The worst experimental design is actually just my samples of disease of interest and experimental control, because unless you have some other diseases that you can pull into this, and this will come back to the broadening of the concept, then you might simply just find molecules that have gone up as a result of sick person, not the disease. So we critique the design of the experiment and make sure that the experiment is a good one to give the samples out to on the hypothesis.
Oh, you don't? Okay.
No. We merely draw upon our experience and our advisors to make sure the experiment is as well designed as possible, because we want the best quality data coming back into the database to enable the next experiments.
Okay. Over there?
Can you say something about the communication that goes on through this 21st Century MS Cafe? Is this going to be all monitored or run through your organization, or is this going to promote the opportunity for people to talk to each other in the cloud through your network?
We're providing a vehicle.
The architecture of the communication.
Yeah, we are not controlling the communication. I mean, there will be some oversight of things that you wouldn't want in that area, but it's our intent to create a vehicle for communication and a source of information, and not much more. I mean, we're not going to be in the business of posing a lot of questions. That's not what we're about.
I'm just wondering about, is there an optimal architecture for the communication? And I'll put this question to the gentleman from Innosight as well.
It's an interesting question.
Other models out there that might lend themselves to this discussion.
Well, and as a follow-up to that as well, I was going to ask you your perspective on how the MS Discovery Network will exist as a complement to, say, Patients Like Me, which we discussed. And there's about 25,000 self-diagnosed or at least self-identified MS patients on Patients Like Me. How do you see the two similarly or differently?
So actually, I think they're completely different, because the MS Discovery Forum is for people who are interested in doing research in the field. It's not a place for patients to share information. That being said, one of the members of this new disease bio-network organization is Patients Like Me. And in fact, going back to the information that you might want to be including in your model, self-reported patient information is something that everyone is becoming very excited about.
Back to the job to be done, do you have a series of jobs that are important and unsatisfied enough by the major social networks to create these more focused communities and actually have them be successful?
And I should say, the Multiple Sclerosis Discovery Forum is not a unique idea. It's a implementation of an idea for multiple sclerosis. The same group, the Mass General Institute for Neurodegenerative Disease, created in the last year the Pain Research Forum, and there also is the Schizophrenia Research Forum and the ALS Forum, an Alzheimer's forum. So these fora are special interest groups that want to create worldwide threads of discussion around particular areas to generate better ideas and to access resources is not completely novel. It just hasn't existed in the multiple sclerosis area before. And putting it together with a biorepository is definitely new.
Right here?
So to that point, would you say that your perfection of the process that's associated with the accelerated curve could then be taken into other disease states, and you should accelerate into those other disease states as the preferred perfected model? And you already, to the point of the woman in Florida, you already have a grant, right? The accelerated cure. So.
Actually, that's a very good point. I think Linda Cann, a board member, and I have already begun that sort of discussion as to whether, you have to be careful about focus, and you have to be careful about resources. However, that having been said, and I'm going to be on a hobby horse for a moment here, one of the things that really disturbs me is the proliferation of institutes at the NIH. So the NIH is a major funding body for biomedical research. I went to the NIH in 1976 as a postdoctoral researcher. The institute I joined was an institute whose initials were NINCDS. It was the National Institute of Neurological and Communicative Disorders and Stroke. Well, a few years earlier, the institute had been NINDS. And a few years before that, the institute had been NINDB. So let me start with the history at NINDB. That was the National Institute of Neurological Disorders and Blindness. And then there was a lobby within Congress to create an institute specifically for people with vision problems. And so they created the National Eye Institute. And the B came out and was replaced by an S. By the time I got to that institute at the NIH, the C had been put in. And that was because there was a lobbying, congressional lobby, about hearing disorders. And the communicative was referred to hearing disorders. Well, that C disappeared, and the institute is back to NINDS today. And there's a hearing institute. And on the mental health side, there was a National Institute for Mental Health, and very shortly after there was a National Institute for Drug Abuse, and then there was the National Institute for Alcohol Abuse. Alcohol's not a drug, it's a legal drug, as opposed to the other thing. So we want to separate these institutes. Well, the point is that these institutes are being separated on the basis of symptoms, not on causes and mechanisms. There may be only two ways that a nerve cell can die. And they may be dying that way in Parkinson's disease, multiple sclerosis, Alzheimer's disease, which, by the way, is in the National Institute of Aging. And so, actually, there might be a reason to create the ACP of neuropsychiatric disorders. And that actually may be what One Mind is heading towards, and we may be able to help them facilitate that. But it's a very good question. It comes down to focus and resources, but actually there's a scientific advantage to bringing a lot of the neurological and psychiatric disorders together, because we don't know very much about any of them. And who's to say that epilepsy, which resides in the National Institute for Neurological Diseases, isn't actually very similar to bipolar disorder, which is epilepsy of the frontal cortex? Well, that's in the National Institute of Mental Health. And certainly, as the incoming president of the Society for Neuroscience said to me when I was at a meeting and started spouting off like this recently, and I sat back down to him, he shook my hand and said, yeah, I think neurodegeneration in glaucoma is like Alzheimer's disease. So I think actually to bring some of these disorders, which may have very similar mechanisms but different symptoms, together so that we can study them in my disease versus all of your similar diseases, so we really find out what the mechanism is of my disease, not a sick person, is an enormously important thing, and I'd like to push forward to that, but we have to be mindful of resources and focus.
Well, sorry, just to that point,
But if you have $100 million,
you took the words out of my mouth. You get the resources, but the fact of the matter is if you have the perfected or nearly perfected model that says that this works,
Yeah.
then you can raise those resources. And Alex, I wanted to ask you, if you take a look through your experience of models of innovation, have you found ways where people have been able to come together from all these different vested interests to come together to have something more collaborative that could be applied into the medical space?
Yeah, well, there's really three archetypes that we look at of sort of organizations these days. There's sort of solution shops, which are when you really have a complex problem, and it's most of the way healthcare is aligned today, which is bringing together great brains to solve problems that have no real set path to solving them. Then there's what we call sort of value-added process organizations, which just sort of stamp it out like a manufacturing plant, where the outcome is guaranteed every time. And then there's exactly what we've been talking about, which is these facilitated networks, which the internet has made possible, where again, it's really about the process of just being sort of the intersection, enabling communication amongst whoever they are, your partners, your suppliers, your customers, et cetera, et cetera. So certainly the model exists. The model, I mean, we just mentioned Facebook, LinkedIn, all these things are facilitated networks. And that's exactly what ACP has set up. And so the opportunity to potentially stamp out facilitated networks to address these different conditions certainly could exist. I mean, it seems quite plausible.
and I should say that One Mind for Research has had 3 meetings so far. It had the kickoff meeting here in May in the Sheraton Hotel. There was a small meeting in Jackson Hole in July, and the next meeting is on Saturday at the annual meeting of the Society for Neuroscience. And I'm not shy about going to these meetings and speaking up on this topic, and it's actually getting some traction.
What kind of skill set do you have to have in your organization to actually run something like this? And you talked about looking at the tests to see the mechanism, the way the research is going to be done, and you have some people that have some background on that. But then when the information comes in, is there anything you look at, or your organization?
So as I said before, we have quite a strong suit of high-tech folks associated with the organisation. In addition to the internal people that are just so dedicated. I mean, that's the first thing. Have an internal group of people that are incredibly dedicated, right? The skill set is less important, I think, internally than the dedication. But that having been said, the number of people with all sorts of interesting IT backgrounds that are associated with ACP has some real benefits for the organisation. So, for example, we think a lot about data provenance. So that data that came from a group that ended up in our database, what's its provenance? Did someone do a sort on an Excel spreadsheet on one column and screw the whole data? So how do we track back and that sort of thing? So the whole area of data provenance and the data basing and all of that stuff is something that we spend a lot of time with. Unfortunately, we have a lot of people from various parts of the community, from companies, from MIT, that help us in that area. We don't have an internal staff of pure biological scientists, but that's what we're trying to enable outside. So the most important things is that we do a very good engineering type job, and it was founded by engineers that thought that engineers could make an impact on biomedical research. A very good engineering job of putting together the best database, the best biorepository, doing everything to the highest quality standards so that we can enable people with the best materials. And that we have a lot of skills inside.
Well, when I first met you this afternoon, you were describing
Have I changed the story?
No, we'll keep the confidential part.
Okay.
No, public, when you were telling the story basically, I was thinking about, the early days of Linux and the issue of the creation of the open operating system that many of us experienced 10 years back. And I guess to me, what came to mind is, in terms of this information sharing, one of the challenges for some open sources is sort of the self-management and maintenance of it. I mean, Linus Torvalds, when he created Linux, it started with him, but it didn't end with him. He created a community of maintainers, a group of people that would commit over time to keeping this human community functioning, so that you had certain kinds of roles that you could count on being played. And in the case of at least Linux, these were unpaid voluntary roles. The rewards were recognition, doing something valuable. There was, it was, as many of us may know, this was looked at with great skepticism 10 years ago. Why would anyone be doing software coding? Why would anyone be doing patching for nothing? And yet, 10 years on, I think Linux is still with us and its value has been shown. Do you see any concerns with creating the human community of maintainers of the Accelerate Cure project that can keep it going, and there people can volunteer their time and talent to do the important things?
The point is they want to come, and there's something that happens in these types of communities, and that is the people who want to contribute also want to be around the other people who want to contribute. And we haven't quite got to the point where, say, for example, the R community has got to in statistical packages or Linux or any of those things. But I think it's a concern. It would be great if through the Multiple Sclerosis Discovery Forum we could create on the side the support community. That it would be not only the website for the researchers, but it would be the website where there were pages for the support community as well. So that's a really good idea, and I'm stealing that one.
Other questions? In the back?
I have a question just around patient consent and the access to the samples. I mean, I can imagine that, in a typical scenario, what clinical trial you're, partaking in, you sign a consent, but something like this, is it kind of one broad consent form for any kind of research that you're giving out the samples to? How does that kind of setup work?
Yes, the answer is yes to the latter question. We have a very broad consent form. We spend a lot of time providing our clinical sites with material about ACP and its mission that patients can to read. We have quite a lot of patient. We're not a patient advocacy organisation, but there are a lot of patients that follow us in various media. We have a newsletter that goes out. We have Kelly. What are all the things we have?
Every type of social media.
We have every type of social media. We have, in fact, my biggest fear is that people are telling me that I should start blogging. and I'm much more receptive to this than I've ever been. So we ask patients to sign a very broad, this sample can be used for anything, and they sign without coercion and with no payment. And so it's the truth is that the patients are really enthused about it. And it's a completely different activity. I ran clinical trials for many years. Recruiting for clinical trials can be a pain, but we don't have inclusion-exclusion criteria. We are an all-comers naturalistic. We want the samples obtained in a natural treatment setting. So it all comes down to how much you provide information and support to the patients without being a National Multiple Sclerosis Society that spends 70–80% of its money on patient support, which is a jolly good thing, but we provide a little bit of patient support and involvement. And actually we have, I would say that the events that we run, so we just associated with our annual symposium was our annual gala dinner, at which our chairman of the board was honoured for his wonderful work. The reason to have events is, yes, to raise money, but it's actually to provide the awareness and to build the community, to make it possible for people to understand what we're doing and to sign that all-encompassing release form.
You sounded a few minutes ago like you put a great deal of thought into your theories on neuro and psychiatric disease. Or are those just like cafe thoughts, to quote you, or is that something you think you might actually pursue, like an ACP for neuro and psychiatric disease?
It's too soon. It's like the, I think it was, who was it? It was someone asked Chiang Kai-shek what he thought of the French Revolution, and he said it's too soon to tell. It's probably the right answer as well. You know. So I think it's too soon to tell. I think that I'm going to continue to go to the One Mind for Research meetings as that organisation develops, and if it develops, it may be possible to create a whole ACP type organisation underneath the One Mind for Research umbrella. I've been involved in research in neuropsychiatric diseases in one form or another for a few years, you know, one of the things that amazes me is how similar in many ways a lot of these disorders are. And I mentioned, I think it was Colette mentioned the protein that my former company Cambridge Neuroscience discovered that was a potential treatment for multiple sclerosis and still is being pushed forward by another company these days. So that protein is called neuregulin-1, and we discovered it because we were interested in stimulating nerve regeneration in the peripheral nervous system to stimulate repair following damage. And then it was discovered that actually if you took that protein into a model, an animal model of multiple sclerosis, you could actually affect the course of that experimental multiple sclerosis. And we went, well, that's really interesting, but that's not so different because we're dealing with nerves here. And then a company called Decode Genetics out of Iceland discovered that this neuregulin-1 gene had an enormous relationship with schizophrenia. You could have knocked me off my socks. But I think what it proves is that we actually don't know much about the causes and mechanisms of these diseases. So knowing that this protein was involved in myelination, which is the sheath around nerves, I Googled schizophrenia and demyelination, and lo and behold, 2 or 3 papers come up that I'd never heard of. You know, and so maybe there was a reason already to think that this might be the case. So we just don't know how close these different disorders are. And in the case of multiple sclerosis, it's a degenerative disorder of the brain. So depending upon where it grabs hold of the brain, you can have neuropsychiatric symptoms, or you can have motor symptoms, or sensory symptoms, or visual symptoms. And so I think it's very good to keep an open mind about how you might push Accelerated Cure Project forward.
Okay, that's the second question. This is a follow-up on our lunch. You had mentioned in your comments at lunch that in these kinds of neurodegenerative disorders, a person might have a condition for 10, 20 years, but it's only until the reserve of redundant neurons are depleted that one can actually detect. So that, to me, the question is there work in terms of ACP towards early detection? In other words, finding out earlier in the phase before massive loss of neurons has occurred.
You should have come to our symposium on Saturday weekend because actually, as the assembled physicians were talking about something that would be transformative that ACP should work on, it would be to find markers that were indicative of neurodegenerative processes in an early stage multiple sclerosis patients, because those markers would then be something you could develop drugs against. Because just like, what I mentioned was In most neurodegenerative diseases,
leading aspects of private life sciences companies here in the Boston area. What motivated you to take this assignment on at this stage in your life? That's question one. Question two is, now that you have the job, what are some of the daunting leadership challenges that you see for the organization as you help it achieve whatever you hope it does?
okay. So first one is that I'm only 16. So I have a substantial career in front of me, and this is just such an innovative idea that as I said, it was compelling, and you know, I'm not old. I'm young. So,
Nor did I suggest that.
so it's just, it is, in a way, it's an opportunity to spend some time exploring this model and making an impact in a disease area of substantial importance that I have an affinity with, if you like. I don't have anyone in my family that has multiple sclerosis. I'm not motivated like that. I'm motivated because it's an important disease area, one that I've worked on in part in the past, and this is a very innovative approach. And I've done the drug discovery and development thing. I could do that again, but actually this and a company I started 2 years ago that's focused on clinical decision support systems in psychiatry are the sort of things that I'm really interested in now. So once upon a time I used to study the behavior of single proteins in a single nerve cell. Now I think about healthcare. And so it's an evolution, and how do you change healthcare? So it's evolution. With regard to the challenges, one of the leadership challenges that I have is to understand the world of not-for-profit fundraising. You know, my usual approach to fundraising in a biotech company has been to devise a project and then either go to investors or management or to a pharma company and get that project supported and moved forward. Project has a very clear relationship between the objective and the deliverables. I don't quite understand yet how to deal with, although it may seem like I'm rather confident about what we should be doing, I don't really understand all of the ramifications 4 months into the job of what it's really going to take to move Accelerated Cure project forward. I have plenty of vision, and the people inside ACP have plenty of vision, and the board has plenty of vision. But I think implementing the vision in an efficient way and supporting it with resources is a real challenge. I don't think that there's any challenge with getting people enthusiastic about what we do, whether it's researchers or clinicians or maybe the general public. But how you turn that how you turn that enthusiasm into support is something I really think is a challenge at the moment, because I have no experience in that area. So why on earth did you hire me?
Robert, would you say the same thing with regard to the dynamics of innovation within the for-profit world and not-for-profit, that it may be a little too early to tell whether it's easier or harder in one area or the other?
So Jamie Hayward, one of the founders of Patient Like Me, and I had a conversation because we interact quite frequently. And Jamie, of course, founded an ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease, foundation based upon his brother's disorder. And you've probably seen the PBS series and maybe read the book. And then he went and founded Patients Like Me. So the ALS was a non-profit, the Patients Like Me is a for-profit. And we had a conversation. So every time he sees me now, he says, "How's it going?" And I said, "It's going fine. Things are really great. We're pushing forward. There's great success, plenty of proposals coming in. You know, we're getting very excited with the opportunity to form the clinical network, and he's part of this bio network." So I say, "This is a really great opportunity for collaboration." I said, all of this is really going great, but I don't completely understand the non-profit world. I don't completely understand. He said, oh, it's very simple. He said, in a startup company, there's an exit strategy. So I actually don't think there's a lot of difference in, except for the thing that most companies focus on, as I said, most companies focus on a specific product goal. And we have a product goal, but it's not quite the same. It's not quite the same as what I'm used to. But in general, I think that, you know, motivating people to do stuff is pretty much the same.
Yeah, and quite often, as we see in for-profit businesses, there's such the profit imperative and such the core business imperative, which just drives everything the company does. And anything that doesn't look like the core business becomes made like the core business. We sometimes call it sort of dogging the cat, right? The dog makes the cat look like a dog. So perhaps you'll find that that's a little bit different in this organization.
Maybe. I don't think I've been there long enough to really understand everything.
One more quick question?
Yes, I've had the microphone for a long time, so I'll ask the question now. I was curious about the samples. You know, the patients are all over the world. How do you collect samples? How do you maintain them? And what's the shelf life of the samples?
Okay, very good questions. The patients are not all over the world. The patients are all over the United States. And the samples are collected and shipped to a commercial organization called CICL in Maryland that has all of the facilities and does much more than storing our samples. We're a small component of what CICL does. It's a very professional commercial organisation. The samples are sent by shipping, by freight, by Federal Express, to all parts of the world, and there's a well-established system for sending biological samples, frozen or otherwise, to the four corners of the world. So that all of that is done absolutely at the top level of professional commercial organisations. We have not collected any samples from outside the US yet. One of the discussions that I had with the president of the International Federation of Multiple Sclerosis Societies was whether actually ACP could go global and what that would mean in terms of sample collection, because we're only tapping into a very smallish number of centres and samples. And the truth is that the work in this area really is enabled by large numbers of high quality samples.
Robert, thanks very much. Bob Collette.